Okay, this week we're going to talk about cell communication. So there are two main different types of cell communication. There's intercellular communication and intracellular communication. Inter means between, so this is communication between cells. Intra means within, so intracellular signaling is communication within a cell. Signaling cells will release chemical signals to target cells that are affected by these chemical signals. Signaling cells are generally termed ligands, so they're a molecule that binds another specific molecule, and it sends a signal to a target cell, also known as a receptor, which will be affected by the signal sent by the ligand. There are four main types of signaling: paracrine, endocrine, autocrine, and direct signaling. And it's important to note that not all cells are affected by the same signals. They will definitely differ among different types of cells, among different types of tissues as well. And the main difference between these four types is the distance that the signal has to travel to reach the target cells. In paracrine signaling, these are signals that are acting locally between cells that are close together. They will move by diffusion, and this will elicit quick responses over a short period of time. Ligands that work by paracrine signaling are quickly degraded for quick diffusion if more are released. So this results in a very high concentration of ligands. An example of this is signals across synapses. So that happens within neurons. The junction between nerve cells, or neurons, is called a synapse. So the synaptic signal then would be the chemical signal between the nerve cells. The ligands that are performing this chemical signal are called neurotransmitters. With endocrine signaling, this will be very much the opposite of paracrine signaling. So with endocrine signaling, endocrine cells will send out chemical signals to distant tissues, and these generally have a slow response with a longer lasting effect. So in contrast to with paracrine signaling, the neuronal communications, which had a fast response with a short lasting effect. The ligands with endocrine signaling are called hormones, and they travel through the bloodstream and tend to be in a diluted or lower concentration. With autocrine signaling, these are produced by signaling cells that also bind the ligand that is released, so they are performing the signaling on themselves, and this generally occurs during early development of an organism through the embryo. This is a way for the cell to check itself to make sure that it's functioning properly. It also helps to regulate pain sensation and inflammatory responses, and if things are not functioning properly, or the cell notices there's a virus or a bacterium that's invaded the cell that's causing it to not function properly, the cell will undergo apoptosis or programmed cell death. There's also direct signaling across gap junctions, or in the case of plants, plasmodesmata. These signaling molecules that diffuse between cells and gap junctions or plasmodesmata are called intracellular mediators. And because plasmodesmata in plants are so ubiquitous, this results in the plant just being a giant communication network, all the cells being able to communicate with each other. So in this way, a group of cells can coordinate response to a signal only one of them received. It's kind of like a really long game of telephone. Receptors are protein molecules in a target cell that bind to the ligand, and there are two main types of receptors: internal receptors and cell surface receptors. Internal receptors are also known as intracellular or cytoplasmic receptors. Therefore, they are found in the cytoplasm of cells. They respond to hydrophobic ligand molecules, and they tend to act as regulators of mRNA synthesis. So in this way, they directly influence gene transcription. With cell surface receptors, these are also known as transmembrane receptors. because they are found on the cell surface and they are anchored into the membrane as integral proteins. They bind external ligands and they are very specific to individual cell types. Because they are integral proteins, malfunction of them can result in some pretty significant diseases such as hypertension, asthma, heart disease, and cancer. And there are also 3 different types of these cell surface receptors: ion channel linked, G protein linked, and enzyme linked. In ion channel linked receptors, these receptors will bind a ligand, which will induce a conformational change or a shape change within this protein. A channel will open through the membrane and allow only specific ions to pass through. In G protein linked receptors, they bind a ligand and activate a G protein, which will then interact with an ion channel or enzyme to allow substances through. Some pathogenic bacteria can actually release poisons to interrupt G protein functions, which will result in diseases such as pertussis or whooping cough, botulism, and cholera. In enzyme-linked receptors, the intracellular domain is associated with an enzyme. The signal travels through the receptor and will activate the enzyme, so then the enzyme will perform its function. So again, the signaling molecules, another word for them is ligands. So we have 3 specific types of ligands: small hydrophobic ligands, water-soluble ligands, and other. With the small hydrophobic ligands, they diffuse through the plasma membrane and interact with internal receptors. Some examples of these are steroid hormones such as estrogen, testosterone, or cholesterol, thyroid hormones, and vitamin D. So in order for them to be transported through the bloodstream, they have to bind carrier proteins. With water-soluble ligands, they can't pass through the plasma membrane by themselves through diffusion like the hydrophobic ones can, so they have to bind to cell surface receptors. Examples of these are smaller molecules, peptides, and proteins. And one specific in the other category that we want to talk about is nitric oxide, or NO. It diffuses across the membrane and induces relaxation of smooth muscle tissue. It causes blood vessels to dilate, which is why it's implicated in the mechanisms of nitroglycerin and Viagra. Signal transduction is the continuation of cell signals through the membrane and into the cytoplasm. So this only occurs with cell surface receptors, and it often occurs when 2 receptors bind together to form a stable complex, called a dimer, which in the process is called dimerization. So activation of the receptor's intracellular components when the ligand binds induces a signaling pathway or cascade. In different parts of the pathway or cascade, things that are above it in the pathway are called upstream events, and things that are occurring later on in the pathway from a certain point would be called downstream events. Signal integration is when signals from 2 or more different cell surface receptors merge to activate the same response in cells, and these signals can be amplified via enzymatic cascades. So there are 2 different ways that enzymes can amplify these signals: phosphorylation and second messengers. Phosphorylation, as we've talked about before, is the addition of a phosphate group to a molecule. Generally, this is catalyzed by the enzyme kinase. And usually the enzyme is named after the substrate it's phosphorylating. There are some exceptions to this rule. Sometimes the kinase will be named after the product if this is in a reversible reaction. Phosphorylation may activate or inactivate an enzyme, it depends, but regardless, dephosphorylation of the same molecule will have the reverse effect of the phosphorylation. Second messengers will propagate a signal after it has been initiated by binding of a signaling molecule or a ligand to the receptor. So this will help to amplify whatever signal is being made by the ligand. Two good examples of second messengers are calcium and cyclic AMP or CAMP. Calcium amplifies signals by flowing into a cell, as generally the extracellular fluid has a higher concentration of calcium than the intracellular fluid does. So the influx of this calcium into the cell causes a lot of different amplifications of signals within the human body. Two specific examples are the release of insulin in the pancreas and muscle contractions. Cyclic AMP or CAMP binds and activates CAMP-dependent kinase or A kinase, and that's important in a lot of our metabolic pathways. Another example of second messengers are inositol phospholipids. These are located near the membrane-bound receptors. So two examples of inositol phospholipids are diacylglycerol or DAG, and this will phosphorylate amino acids, and inositol triphosphate, which will propagate calcium signaling. Some signal transduction pathways will regulate transcription and translation. So this will directly affect gene expression. The MAP kinase ERK or MAPK-ERK pathway regulates translation, and PKC inactivates the inhibitor of transcription to make transcription possible. The book goes into a lot more detail on these two pathways. I don't care that you know the mechanism, just what they regulate. During fight or flight mode, when your adrenaline is pumping, you have beta-adrenergic receptors in your muscle cells that are activated by adrenaline, also known as epinephrine today. When these receptors are activated, it breaks down glycogen, your storage form of glucose, into glucose, and it inhibits the formation of glucose into glycogen. So you have a lot of glucose freely floating so that you can use it during this fight or flight mode as just an extra pool of energy. Generally, cells don't divide unless they're stimulated by other cells to do so, specifically stem cells. So ligands that promote growth are called growth factors. Initiation of the growth factors results in cell division. Cell death, or in the programmed sense, is apoptosis, like we talked about earlier. And apoptosis is really important so that we have properly functioning cells in our body. Sometimes T cells, a type of white blood cell that you have, will target your own cells instead of foreign invaders like it's supposed to, which can result in autoimmune diseases. Apoptosis is also really important with embryonic development. It gets rid of a lot of the structures that we have in utero that we no longer need as out of utero, such as the webbings between fingers and toes. So if you are born still with that webbing between fingers and toes, that's a failure in apoptosis. There's three main ways to terminate a signal cascade. If you degrade the ligand and remove it, you no longer have that stimulus present for the receptor. Phosphatases are the opposite of kinases, so instead of phosphorylating something, it will dephosphorylate something to remove the amplification of that pathway. And also phosphodiesterases, they degrade CAMP, so this will decrease your calcium signaling. So signaling pathways also happen in single-celled organisms. So it happens in yeast. Budding yeast secretes a mating factor, which will bind to nearby yeast cells, telling them to stop their normal growth cycle and initiate signaling cascades to prepare for reproduction. In bacteria, this is called quorum sensing. This was first found with the symbiotic relationship between bacteria and the Hawaiian bobtail squid. So if there's enough bacteria present and active, they will allow this squid to bioluminesce. So scientists found out that bacteria can actually tell how many other bacteria of the same species are around them. So that's how they came up with the name quorum sensing. Quorum is, in politics, the number of members required to be present to vote on an issue. So in this way, bacteria can sense the amount of bacteria of their same species. So in the case of bacteria, instead of ligands, their signaling molecules are known as autoinducers. These autoinducers will bind to transcription factors, which can turn gene expression on or off. Autoinducers will increase with the number of bacteria, which is a positive feedback loop. So the more bacteria there are, the more signaling there is. So that's how bacteria are able to communicate with one another to allow things like bioluminescence to occur.